glucose concentrations during a day. Avoid the 12-time measurements at a day.
More questions, comments? Thank you very much again. And we move to the last presentation of this session by Anjaparavanda Naren from the Cystic Medical Center. Dr. Naren, can you hear us?
Yeah. Can you hear me?
Yes.
Is the presentation okay?
Well, you should
swap presenter.
Yeah.
He told me to tell you.
I can see.
Is it okay?
Yeah.
Perfect.
Does it look okay now?
Yes.
It's working.
Thank you. First of all, let me congratulate Peter, the Hungarian Pancreatic Society, for an incredible job. And I want to thank Peter Mikulas and the organizing committee for having invited me, and Mysom for having introduced me to the committee. I'm honored and humbled to be presenting to this incredible group. And I hope I don't screw up, okay? So I have nothing to disclose. And this is the God molecule in the lab. It's also called cystic fibrosis transmembrane conductance regulator. It has 2 nucleotide binding domains. And in this nucleotide binding domain is an ATP binding cassette, and therefore the name ABC transporter. The cryo-EM structure is shown on the right. And the point I want to make is the unique thing is this regulatory domain. It's the phosphorylation, PK or PKG dependent phosphorylation of this domain that determines whether the channel opens. So it's, as I told you, the PK or PKG dependent phosphorylation of this domain allows chloride to come out. As chloride comes out, sodium follows it. It creates an osmotic driving force for water, and that's how it regulates the transporter. And if there is little or no activity, it causes cystic fibrosis. Too much activity, that causes secretory diarrhea. Now, this audience is very aware. What of interest to this is the presence of CFTR, heavily present in this particular structure, the exocrine pancreas, especially in the duct. The CFTR is present all along the ductal lining. And yeah, it's a chloride channel. And as it moves salt and water across, it allows the acinar cells, which makes the digestive enzymes to move towards the duodenum, where it can digest the enzyme. If the duct gets clogged in cystic fibrosis, you have pancreas, which are kind of dilated, and calcification and fibrosis happens. And that leads to pancreatic insufficiency, which is the most common symptom among CF patients. And with time, CFRD happens, which I'll get to in a second. So there are more than 2,000 mutations described in CF. I won't go into great detail about this, but I just want to make a point. There are several classes. They can be classified into several classes. The class 2 is one of the most common. 70 to 90% of the patients have at least one allele with this particular class. Delta F5 being the most common. Trikafta is a drug that is FDA approved currently. This is a folding defect. It goes to the ER. But I also want to highlight class 3. It goes to the plasma membrane. This was the first drug, Kalydeco, that was discovered for this particular class of mutation. So here is a clinical case I want to talk about. This was one of our first patients back in Memphis. It was F508 in one allele, S549 in the second allele. At that time, we did not know what this mutation was. She was diagnosed as CF in 5 months. She was below the 5th percentile of all growth. She has high sweat chloride in both arms, pancreatic insufficiency, pseudomonas by age 4. She has multiple hospitalization, FEV1 of 50 to 60% predicted. And this is what a CT scan looked like, severe fibrosis. And this is what the typical tree in bud opacity looks like. So we looked at the, as I told you, in the ATP binding cassette, there is a signature sequence. It's LSGGQ. The G551D is right here. And near that is S549 region. And this S549N is kind of popping out. And so we went ahead and made the mutation there, and it was completely dead. The function, there was no function for this particular protein. But what was interesting was this protein was very similar to G551D. That is, it comes to the surface, but it is, and that is shown here as well, surface labeling, and it is non-functional. What was cool was we made lentivirus vector, and then we transfected cystic fibrosis bronchial epithelial cells and went ahead and in Wusin chamber, when we challenged it with forskolin, we didn't see a response until we challenged it with ivacaftor or VX770, we saw a beautiful response. Then we basically proposed that these the patients would respond to Kaleidocope. Then as we moved to Cincinnati, we had this UAE patients. This was a first cousin. I just had to remove the picture. I wasn't sure if I was allowed to show the picture here. So these were first cousins. It was a consanguineous marriage. So it was an open label study. We did an N-of-1 clinical trial. All 3 children were S549N mutations. So the twins, they were 11-year-old, and the older boy was 16. So the question was, would they respond to ivacaftor? So I'm just showing the older boy's data. I'm not going to show you everything. I'm going to finish this in a couple of slides. He responded very dramatically. The sweat chloride dropped dramatically within 16 days. The FEV1 increased as expected. But of interest was this study, the 2 girls did not show an impaired oral glucose tolerance test. The older boy, however, the 16-year-old, showed an impaired oral glucose tolerance test, whereas at the day 31, the oral glucose tolerance test seems to kind of come back to normal. But what was exciting was this study in which we saw the insulin secretion increase dramatically by nearly 5-fold when the kid was on Kaleidocope. So this kind of was an important study because, as you see, CFRD increases with time, as shown. The arrow basically points the risk factor. As the children get older, the CFRD increases, the risk increases. We were able to catch this boy at the age of 16 around this time point. And the reason this is important is children who get CFRD are 3 times more likely to have mortality compared to children who do not have CFRD. So the question we asked is there cell-cell signaling between pancreatic ductal epithelial cell, that is the exocrine pancreas, and the islets, okay? So we were working, these are the kids who developed all these model systems, and I owe a debt of gratitude to them. We were working with the mouse model systems, the mouse trachea, the mouse enterosphere system, but we were not able to generate any data from there. The exocrine pancreas, we were not able to generate any data from there. So we developed our model systems. In Cincinnati, we were able to develop the model systems from the human. And we developed, as you know, the CF is a multi-organ disorder. We look at the trachea, the lung on a chip. In the pancreas, this is what the topic of discussion is, the ductal organoids and pancreas on a chip. And we also do the intestinal model system as well. So the question we asked is, can we generate a CF pancreas? And of course, you can do it. All you need to do is recruit a smart bioengineer. And Kushik, I don't know why he joined my lab, but brilliant bioengineer he is. And the question was, do we have a microphysiological unit representing a living pancreas, reconstitute 3D microarchitecture, mechanical activity, epithelial islet interface, monitor CFTO function? So this was a beautiful collaboration with Jamie and Myson. They're in the audience there. I owe them, again, a ton of gratitude. Jamie, in fact, after his surgery, he was kind enough to even sometimes bring the pellet and make sure we had it with us. And I'll not go into these details about this, but the point I want to make is the pellet has the acinar ductal cells and the islets. And what Kushik had the ability was surgically isolate these ducts, very beautifully isolate them. And from these ducts, we were able to isolate these ductal organoids. And the first thing we did was did RNA-seq experiments to show that these were indeed highly purified components, and they were devoid of any blood cells or acinar cells or islets. And importantly, they had all the epithelial biomarker using immunofluorescence as well. But of point of interest is CFTR showed beautiful apical staining. This was the antibody that Chris Marino had developed. And when he was in Memphis, he allowed me to have a certain amount of antibodies when he was shutting down his lab. And one of the first things we did was we polarized them and made sure this is the gold standard, basically polarize them and use forskolin to show CFTR function, and then use CFTR inhibitor to show that you can inhibit the function completely. And when we were doing this, we also isolated the islets from these remnants that Jamie gave us, and we were able to show that these islets were, had insulin and glucagon as well. And then they were functional, low glucose and high glucose. And what Cystic did was, he's being a bioengineer, he decided he's going to build a chip. And this is Don Inberg had done this in Weiss Institute on lung-on-a-chip, published in Science a few years ago. So we kind of modified it to kind of make it look like a ductal chip, which has kind of these branched structure to mimic the duct. So Cystic developed this chip, and he made me hold it, and my name is here on it. When we were building this, the technology transfer office wanted to know if we had any ideas of making any kind of technology transfer and intellectual property. So we had some ideas. Those of you who are familiar with the Seinfeld character, Jason Alexander, George Constanza, protects his hand when he becomes a hand model. So we had ideas about that. And we also thought maybe we can make t-shirts about it. And we have some other ideas as well. But seriously, why microfluidic devices? So they cost less than $2 per chip. They're highly reproducible, miniature tissue feed cells automatically, monitor cell function in real time, variety of applications, highly sensitive. So one of the first experiments we did was we isolated the ductal cells, put it on a chip, made sure they're functional. It's a very CFTR-centric lab, as I told you. We did a efflux assay, and in the same chip, we also made sure that, separate chip. We made sure that we could see that the islets were grown, and we needed about 15 islets to see this function. And the question then we asked was, can we mimic this cell-cell signaling? That is, can we look at the pancreatic ductal epithelial cell and islet, and can we build a chip? This is, again, the insulin here and the CFTR imaging of the duct here showing the staining using this Chris Marino's antibody. And essentially, we built a double channel chip to kind of have islets on one side, at the basal side, and the duct on the apical side. So that's how we did it. We grew the ductal epithelial cell on the apical side and the islets on the basal side. And we had a pore of 10 micron size. And this is a PDMS membrane that is a smart membrane that is kind of treated with PVA. So it has a hydrophilic surface. And then we basically looked at insulin secretion, and we can see insulin secretion with low glucose and high glucose. And then the experiment was, you basically treat on the apical side, you do a wash, you collect it, you have a wash with CFTR inhibitor with low glucose and CFTR inhibitor with high glucose. And what we saw was, lo and behold, when we added CFTR inhibitor, we saw inhibition of insulin secretion. And now what we did was the same experiment here with the pore. When we inhibited CFTR, we saw that the insulin secretion was inhibited. Whereas when we removed this pore, there was no pore here, there was no cell-cell communication, then you do not see the inhibition. Whereas if you don't have any ductal cells and you either add inhibitor or not, you don't see an effect as well. So the conclusion was that there is cell-cell signaling between pancreatic ductal epithelial cells and islets. And so to conclude, the dysfunction of CFTR directly affects insulin secretion in islet cells. We think we have identified the metabolite responsible for insulin secretion. That's something we are working towards. We are trying to build sensors, chloride, pH, and insulin sensor. We are also trying to build a bicarbonate sensor. We are trying to add another cell chamber. That is, we are trying to have ACE in our cells. That has been slightly a challenge. We can keep it alive for about 7 days with secretion. We are trying to extend its lifetime. We are trying to see the correlation between these 3 cell types. And as you can see, my lab has a very strict dress code, but seriously, these are the young women who helped me set up the lab. I owe them a lot when I first moved to Cincinnati. And these are the people who are with me right now. And they are the ones that make me, that help set up the lab and do all the school stuff in the lab. And I'll be more than happy to take any questions. Thank you.
Very much for great talk and amazing pictures. Is there any question?
May I have a question? Can I ask a question? Okay, first, great talks, really interesting. The questions I have, actually, it's more practical. For how long the duct, the tissue will survive on the chip, and to what extent you can actually manipulate it genetically while on the chip?
On the chip, we have been able to keep it for over a month. As organoids, we've been able to keep it for almost 4, 5, 6 months as organoids. And we can freeze it. We have distributed it to several labs also as organoids. So
would you need, do they proliferate? Do they do anything while on the chip? And they just stay exactly as it is, and they maintain polarity,
Yeah, they maintain polarity on the membrane for about a month. We are able to keep it for a month.
Yeah. So that's long enough to be able to.
Yeah.
Thank
you. Technical. Do you have a problem with bubbles?
Initially we did, but we have found ways to kind of get around it.
Other questions, comments? If not, thank you very much again for the